Welcome back to the deep dive. Today, we are heading into the anaerobic world, these deep, dark places in the body where oxygen isn't just scarce, it is actually a poison.
Mm-hmm.
We're talking about a group of bacteria that I like to think of as the biochemical terrorists of the microbial world.
That's a pretty dramatic description, but honestly, it's not wrong. It's actually, it's entirely accurate. It really captures the core of what we're dealing with here.
Right. We are wading into the genus Clostridium. And these are not your run-of-the-mill infections where the bacteria just invade and multiply and crowd everything out.
Yeah.
These guys, they're playing a completely different game.
They are. They are biochemical warfare agents.
Yes. They don't need to be there in huge numbers to just cause absolute devastation. They just need to get their little toxin factories up and running.
And that is the critical distinction. When we study Clostridium, we are really studying the power of toxins. It's less about the bug invading, though, that does happen, and much more about the chemical weapons they manufacture and release. It is a fascinating and, frankly, a terrifying area of microbiology.
It really is.
And just for our roadmap today, we are basing this deep dive strictly on Murray's Medical Microbiology, the 9th edition, Chapter 30. We'll be pulling in some key supplementary details from Jawet's where it helps to, really flesh out the clinical picture or the biochemistry behind these toxins.
So if you are a medical student prepping for boards, or really just anyone who wants to understand why a rusty nail or a swollen can of beans just scares the living daylights out of doctors, then you are definitely in the right place.
Absolutely.
We have 4 major villains to cover today: Clostridium difficile, perfringens, tetani, and botulinum. But before we meet the whole cast, we really need to understand the stage they play on. So what defines a Clostridium? What makes it tick?
Okay, so there are 3 core features that you absolutely need to lock into your mental model. If you see the word Clostridium on an exam or, on a lab report, your brain needs to instantly fire off. Gram-positive rods, spore formers, and obligate anaerobes.
Let's break those down. Obligate anaerobe. I mean, that means they hate oxygen, but it's more than just a preference, isn't it? It's a metabolic necessity.
It is, yeah. A lot of the bacteria we deal with are facultative. They can take oxygen or they can leave it. But for Clostridium, oxygen is actively toxic. They just don't have the enzymes, specifically things like superoxide dismutase and catalase that other organisms use to neutralize the toxic byproducts of oxygen metabolism.
So if you expose a vegetative cell, the active growing form, to fresh air,
the oxidative stress just tears it apart. It basically rusts from the inside out.
and that right there dictates where they can live. They can't just hang out on your skin like staph might. They have to find a very specific niche.
Exactly. They need an environment that is completely devoid of oxygen to thrive. So we are talking about places like deep, devitalized wounds where the blood supply has been cut off, or the lower gastrointestinal tract where the oxygen tension is naturally very, very low. That's their playground.
Okay, but that seems to raise a huge logistical problem for the bacteria. If fresh air kills them, how on earth do they survive out in the world long enough to infect us? The world it's full of oxygen.
Right.
How do they get from the soil into a deep wound without dying in transit?
And that brings us to their evolutionary masterpiece, the spore.
A bunker.
The apocalypse bunker. That's a perfect analogy. When environmental conditions get tough, like when there's too much oxygen or not enough nutrients, these bacteria kick off this incredibly complex genetic program. They dehydrate their DNA, they wrap it in multiple protective layers of, keratin and calcium to picolinate, and then they just shut down their metabolism completely.
So they go dormant.
Dormant is a massive understatement. These spores are, for all practical purposes, indestructible. They can survive boiling, drying, radiation, most chemical disinfectants. They can sit in the soil for decades or on a hospital bed rail for months just waiting.
So they are bunker busters. They hide out in the bunker, the spore, until they detect some kind of signal that they've landed in a nice, warm, oxygen-free environment, like a deep puncture wound or, say, a gut that's been wiped clean of other bacteria. Then what happens?
Then they germinate. They transform from that dormant spore back into the vegetative state. And vegetative in microbiology doesn't mean it's passive like a vegetable. It just means it's metabolically active. It's a dividing. And most importantly, it's turning on its toxin genes.
Which brings us to our third big theme: biochemical amplification.
This concept is what drives the incredible lethality of this genus. You don't necessarily need a massive army of bacteria to kill a patient. A relatively small number of Clostridium bacteria can produce these unbelievably potent exotoxins that cause systemic effects far, far out of proportion to the actual bacterial load.
Tetanus and botulism are the perfect examples of this.
The absolute prime examples. You might have this tiny, insignificant infected wound, but the tiny amount of toxin produced is enough to shut down the entire nervous system.
Okay, so the stage is set. We have our anaerobic spore-forming bunker busters. Let's start with the one that's probably causing the most daily headaches in hospitals right now, the modern plague, Clostridium difficile.
Or as everyone just calls it, C. diff. And we should probably note there has been a taxonomic shift recently.
No, of course.
The taxonomists who, seem to love making medical students' lives harder have officially renamed it Clostridioides difficile.
Clostridioides.
Right. But functionally, it's still a Clostridium, and clinically, absolutely everyone still just says C. diff.
Murray lists this as the first major pathogen in the chapter, and it's for a very good reason. It is the leading cause of antibiotic-associated gastrointestinal disease. What makes it such a nightmare in the hospital setting?
Well, it really comes down to its physiology and its relationship with our own microbiome. So C. diff is a large anaerobic rod. As it grows, it produces these volatile fatty acids as byproducts.
And that's what leads to the infamous smell test. I've heard nurses and doctors say they can diagnose a case just by walking past the room.
It is unfortunately very true. It has this characteristic barnyard or horse stable smell. Once you've smelled it, you never ever forget it. But the real clinical issue is the spores. C. diff is a prolific spore former. A patient with an active infection is shedding thousands, millions of these spores in their watery diarrhea.
And those spores land everywhere.
Yeah.
Bed rails, the floor, the toilet handle, the nurse's call button.
Everything. And because they're spores, alcohol gel doesn't kill them.
That's a critical point.
It's so important. You can scrub your hands with alcohol foam until they are raw, but you are just moving the spores around. You have to physically wash them off with soap and water. That's why C. diff rooms are always contact plus isolation, which means gowns, gloves, and mandatory hand washing.
But here's the nuance that I think trips people up. Murray says C. diff is present in less than 5% of healthy people. So if I'm a healthy person and I touch a contaminated door handle and ingest a spore, do I automatically get sick?
No, and this is probably the most important concept for understanding the pathogenesis of C. diff. It is a pure opportunistic pathogen. And to get this, we need to use the squatter analogy.
Okay, let's unpack that.
So imagine your gut microbiome, the trillions of good bacteria living in your colon, as a fully occupied apartment building. There are tenants in every single room. They are the police force of your gut. They eat all the available food, they take up all the physical attachment sites on the gut wall, and they produce chemicals that suppress invaders.
It's colonization resistant.
Precisely. So if a C. diff spore comes along and tries to move in, the police force basically says, sorry, no vacancy here. The spore just passes right through you and ends up in the toilet.
So a healthy microbiome is our shield.
Absolutely. But let's say you get an infection, maybe a sinus infection or community-acquired pneumonia, and you take a course of broad-spectrum antibiotics. Murray specifically calls out ampicillin, clindamycin, cephalosporins, and the fluoroquinolones as the high-risk agents.
They're the carpet bombers.
They are. They kill the pathogen that's making you sick, but they also wipe out the police force in your gut. They basically nuke the entire apartment building. Suddenly, there are vacancies everywhere. Now, if you ingest a C. diff spore from the hospital environment, or if you were one of those 5% of people who already had a few spores hiding there,
It sees its chance.
it sees a massive opening. It wakes up from its spore state, looks around, sees no competition, tons of food, and starts multiplying like crazy.
And that's endogenous overgrowth, if it was already there.
Right, and once it reaches a certain density, it starts producing its weapons. The toxin duo, toxin A and toxin B.
Okay, I always get these mixed up on exams. Let's really distinguish them clearly. What is the mechanism of action for toxin A?
Toxin A is an enterotoxin. The way I remember it is A for attracts. Its main job is to act as a chemotactic signal for neutrophils. It binds to the brush border of the gut, gets internalized, and it triggers this massive release of inflammatory cytokines. It basically sends up a flare that calls the immune system to the scene.
So it's calling in the cavalry, but in a really bad way.
In a completely chaotic way. The neutrophils rush in and they release their own enzymes, which causes intense inflammation and disrupts the tight junctions between the intestinal epithelial cells. This makes the gut wall leaky.
And fluid rushes out into the lumen.
Exactly, leading to watery diarrhea. That creates the main symptom. But toxin B Toxin B is the heavy hitter. It's the cytotoxin. This is the one that does the real structural damage.
What's its target?
Toxin B enters the epithelial cells, and it targets the Rho family of GTpases.
Okay, let's get into the weeds on that. Rho GTpases are signaling proteins that control the cell's cytoskeleton, right? The internal scaffolding.
Yes, precisely. They regulate the assembly and disassembly of actin filaments, the steel beams that hold the cell in its proper shape. Toxin B is an enzyme that glycosylates these Rho proteins. It essentially gums up the works, inactivating them.
So without functional Rho proteins, the actin filaments just fall apart.
They depolymerize. The cell's entire internal scaffolding collapses. The cell rounds up, loses its attachment to the basement membrane, and then undergoes apoptosis. It dies.
So you combine the leaky gut from toxin A with the massive cell death from toxin B.
And you get the clinical hallmark of severe C. diff disease, pseudomembranous colitis.
That is the classic board exam buzzword, pseudomembranous colitis. What does that actually look like if we were doing a colonoscopy on a patient?
If you put a scope inside, you are not going to see a nice, pink, healthy mucosa. You'll see these raised, whitish-yellow plaques just plastered all over the colon wall. And those plaques, the pseudomembranes, are biological debris. They are made of dead epithelial cells, fibrin, mucus, and inflammatory cells that have all clotted together over the ulcerated tissue. It's a landscape of total destruction.
And clinically, this patient would have profuse watery diarrhea, severe cramping, fever, and a really high white blood cell count, a leukocytosis.
Yes. And we should probably mention the hypervirulent strains here, like the NAP1-BI027 strain. This particular strain has a mutation in the gene that's supposed to regulate toxin production. It's like the off switch is broken.
So it just churns out toxins constantly.
Massive amounts of toxin A and B. It also produces a third toxin, the binary toxin, which we think helps the bacteria adhere to the cell surface more effectively. These strains are associated with much more severe disease and much higher mortality.
Okay, so we have a patient, they were on clindamycin a few weeks ago, and now they have terrible watery diarrhea and a fever. How do we diagnose this? My first instinct is to say culture it, but I feel like that's a trap.
It is a huge trap. Do not order a culture for C. diff diagnosis. Culture is way too sensitive. Remember, 5% of healthy people and up to 20% of all hospitalized patients are colonized with C. diff but don't have the disease.
So culture would be positive, but it wouldn't mean anything.
Right. It will grow the bug, but it can't tell you if it's actually making toxins. You end up treating colonization, which leads to antibiotic resistance, and it's just bad medicine.
The goal is to prove the toxin is present.
Exactly. We are looking for the weapon, not just the criminal. The modern diagnostic algorithm is usually a two-step process. Step 1 is a screening test for an enzyme called glutamate dehydrogenase, or GDH.
And GDH is made by all C. diff bacteria.
All of them, toxin-producing or not, so it's very sensitive. If your GDH test is negative, you can be pretty confident the patient doesn't have C. diff.
But if it's positive?
If it's positive, it just means the bacteria are there. It doesn't prove they are causing disease. So then you move to step 2. An enzyme immunoassay, an EIA, that specifically looks for toxin A and toxin B. If that is positive, you have your diagnosis.
What about PCR? Everyone seems to love PCR these days.
PCR is tricky. It detects the gene for the toxin. So it's incredibly sensitive, but again, it can be too sensitive. It might pick up the gene from a dormant spore or from a carrier who isn't actually sick. So if you use PCR alone, you risk overdiagnosing. Most hospitals now use that GDH/toxin algorithm to be more specific.
Okay, diagnosis is secured. How do we treat it? We have to kill the vegetative C. diff without causing even more damage to the microbiome.
Step 1 is absolutely crucial. Stop the offending antibiotic if at all possible. You have to stop the carpet bombing so the police force, the normal flora, has a chance to recover.
And step 2?
Step 2 is direct anti-clostridial therapy. The go-to drugs are oral vancomycin or oral fidaxomicin.
And I want to highlight oral.
Yeah.
Why not IV vancomycin? We use IV vancomycin for MRSA all the time.
This is a classic pharmacology question that shows up on every exam. IV vancomycin is a huge molecule. It does not cross the intestinal lining if you give it intravenously. It just stays in the blood. It never reaches the gut lumen where the actual bacteria are living. You have to give it orally so it can travel down the GI tract and hit the C. diff directly where it lives.
Okay, but here's the problem I hear about constantly. The patient gets treated, they get better, and then 2 weeks later, the diarrhea is back. The relapse rate is really high, something like 20 to 30%. Why is this bug so persistent?
It goes right back to the bunker. It goes back to the spores. The antibiotics, vancomycin, fidaxomicin, they kill the vegetative metabolically active bacteria, but They cannot kill the spores.
They're indestructible.
Right. So you treat the patient for 10 days, the active bacteria die off, the symptoms stop, but the spores are still sitting there in the diverticula or hiding in the mucus. If the normal microbiome hasn't fully recovered yet, if the apartment building is still mostly empty, once you stop the vancomycin, those spores just germinate again.
Round 2, which brings us to the nuclear option for recurrent C. diff, the fecal transplant.
Or fecal microbiota transplantation, FMT, if we're being formal about it. I know it sounds medieval, but it makes perfect biological sense. If the underlying problem is a lack of a police force, a lack of normal flora, then the solution isn't more antibiotics, it is to import a new police force.
You are basically engaging in ecological warfare inside the colon.
That's exactly what it is. You take stool from a healthy, screened donor, you process it to remove all the waste, and you instill the healthy bacteria into the patient's gut, either through a colonoscopy or now, increasingly, through capsules. It restores that competitive barrier. The cure rates for recurrent C. diff are upwards of 90%. It is one of the most effective therapies we have in all of medicine.
That's incredible. So that's C. diff, the opportunistic squatter that waits for us to weaken our own defenses.
Yeah.
Now, let's pivot. C. diff is sort of a slow burn. It takes antibiotic exposure and time. But our next bug, it does not have that kind of patience. It is the speed demon of the family, Clostridium perfringens.
C. perfringens, the boxcar bacterium.
Boxcar. That's referring to the morphology on a Gram stain.
Yes. If you look at it under the microscope, you see these large rectangular gram-positive rods with these very blunt ends. They look just like a train of boxcars linked together. And here's a very high-yield differentiator for you. Unlike the other Clostridia, C. perfringens rarely shows spores in clinical tissue samples.
That's weird. It is a spore former, though, right?
It is, absolutely. In the environment, it forms spores. But in the patient's tissue, it's usually so busy dividing and pumping out toxin that it just doesn't bother with sporulating. It stays in that active vegetative phase.
Murray calls this organism the Formula One of bacteria. Just how fast are we talking?
It's terrifyingly fast. Under optimal conditions, in a nutrient-rich anaerobic environment, C. perfringens can replicate every 8 to 10 minutes.
Right.
Compare that to E. coli, which takes about 20 minutes, or staph, which is closer to 30. This means C. perfringens can go from a small contamination to a massive, limb-threatening load of bacteria in just a few hours.
And that explains why the clinical course of gas gangrene is just so explosively rapid. You can literally watch the infection spread up a limb.
Yes. And let's look at its weapon of choice. In the lab, on a plate, C. perfringens shows this very distinct double zone of hemolysis. This is a key diagnostic visual if you ever see it.
Describe that for us. What's happening on the agar plate?
Okay, so imagine a colony growing on a blood agar plate. Right next to the colony, there's this inner zone of complete clearing. The red blood cells are totally destroyed. It's transparent. That is caused by the theta toxin. But then surrounding that inner zone, there is a wider fuzzy halo of partial hemolysis. That is the work of the alpha toxin.
Alpha toxin. Okay, this is the big one. Murray says this is the single most important virulence factor for C. perfringens. What is it actually doing on a chemical level?
Alpha toxin is a lecithinase. Specifically, it's a phospholipase C.
And lecithin is a phospholipid, a major component of our cell membranes. So this toxin is an enzyme that literally digests cell membranes.
Exactly. It cleaves the head group off the phospholipid molecule. It effectively dissolves the cell membrane of red blood cells, which causes massive hemolysis. It dissolves the membranes of platelets, causing clotting issues, and it dissolves the membranes of the endothelial cells lining our blood vessels.
So it's not just killing cells, it's melting the entire tissue structure.
Correct. And this leads to the most feared clinical presentation, clostridial myonecrosis, or gas gangrene.
Walk us through the typical scenario. How does this start?
It usually starts with significant trauma. Think of a soldier wounded in battle with shrapnel, or a motorcycle accident with a compound fracture, or a deep crushing injury at a construction site. Dirt and clothing that contain spores get jammed deep into the muscle tissue.
Yeah, the tissue is crushed.
Yeah.
So the blood supply is cut off.
Right. No blood means no oxygen. You've just created the perfect anaerobic incubator.
The spores germinate, the bacteria start dividing every 8 minutes, and they start pumping out that alpha toxin.
And the toxin spreads through the muscle planes, melting the tissue ahead of the bacteria. This causes massive edema, massive swelling, and necrosis. And because the bacteria are fermenting carbohydrates in the muscle as a fuel source, they produce huge amounts of gas as a byproduct.
And that's the gas in gas gangrene. So if you press on the skin over the wound, it feels crunchy.
Yes, that's called crepitus. It feels exactly like pressing on bubble wrap under the skin. It is an incredibly ominous sign that the tissue is filled with gas from these bacteria.
And the patient is in agony.
Pain that is completely out of proportion to the visible injury is the classic early sign. But here's the most chilling part, and a classic exam question. If you take a sample of that necrotic fluid and you look at it under the microscope, what do you see?
You see the boxcar bacteria.
You do. But what do you not see?
Neutrophils.
You don't see neutrophils, you don't see white blood cells.
So wait, this is a massive raging infection. The immune system should be swarming that area.
It tries to, but the alpha toxin concentrates in the tissue and it lyses the neutrophils as soon as they arrive. It melts the police force on contact. So what you see on the gram stain is this field of necrotic debris, huge gram-positive rods, and a distinct, eerie absence of inflammatory cells. That is a critical diagnostic clue.
How on earth do you treat something that kills the tissue and the immune system so fast?
You cannot treat gas gangrene with antibiotics alone. It's impossible. The tissue is dead, the blood supply is gone, so IV antibiotics can't even get to the site of the infection. There's only one answer.
Surgery.
You have to use the cold steel, immediate aggressive surgical debridement. You have to cut out all the dead muscle until you hit healthy bleeding tissue. And often this means amputation of the limb to save the patient's life.
High-dose penicillin is used as an adjunct therapy, but the surgery is what saves the life.
Exactly. Without debridement, it is 100% fatal.
Okay. Now, C. perfringens has a side hustle. It's not just a flesh eater. It's also a very common cause of food poisoning. But the mechanism here is completely different.
It is. This is the classic reheated gravy or Thanksgiving leftovers scenario.
So what goes wrong in the kitchen to set this up?
You cook a big pot of stew or a turkey with a big vat of gravy. The initial cooking heat kills all the vegetative bacteria, but the spores survive the heat. Then you leave the food out on the counter to cool down slowly for a few hours. As it cools, the spores realize the heat shock is over, and they germinate in that warm anaerobic environment deep inside the gravy.
And they multiply into huge numbers.
Massive numbers. Then you eat the gravy. You ingest a huge load of bacteria. We're talking 10 to the power of 8 organisms. But here's the twist. The toxin is not in the food yet. This is different from staph food poisoning, where you eat the preformed toxin.
Okay.
With C. perfringens, you swallow the live bacteria. When they hit your small intestine, the environment changes. It's alkaline, they're bile salts. This change triggers the bacteria to sporulate again.
So they turn back into spores inside your gut.
Yes, and during the sporulation process inside your gut, they release the Clostridial enterotoxin.
So you're not eating the weapon, you're eating the factory, and it builds the weapon inside you.
That is the perfect way to put it. The enterotoxin then acts as a superantigen. It inserts into the gut membrane, it forms pores, and causes intense watery diarrhea and severe abdominal cramps. But, and this is key for exams, no fever and no vomiting.
Why no fever? That seems strange for a bacterial infection.
Because it's a localized toxin effect happening in the gut lumen. It's not a systemic invasive infection where bacteria are in the bloodstream. It's usually self-limiting. You feel like you are dying for about 24 hours, and then it passes.
Before we leave perfringens, there's one more really weird disease mentioned in Murray. Pig bell.
Ah, yes. Necrotizing enteritis. This is a fascinating intersection of anthropology, diet, and microbiology. It's seen primarily in the highlands of Papua New Guinea, and it involves a specific strain of C. perfringens, type C, which produces beta toxin.
We've talked about alpha and theta toxins. What does beta toxin do?
Beta toxin causes stasis and necrosis of the gut wall. It's incredibly destructive to the intestines. Now, normally, our bodies produce an enzyme called trypsin in the pancreas, which helps digest proteins. And it turns out trypsin is really good at breaking down beta toxin.
So even if we were exposed to this type C strain, our natural trypsin would just chew up the toxin and protect us.
In most cases, yes.
So what happens in Papua New Guinea to cause pig bell?
It's associated with traditional ceremonial feasts. These feasts involve eating huge amounts of pork, which can introduce the bacteria, and this is the crucial part, huge amounts of sweet potatoes.
What is the deal with the sweet potatoes?
Sweet potatoes contain a heat-stable trypsin inhibitor.
Oh, I see the perfect storm forming.
It is a perfect storm. You eat the pork, which introduces the bug that makes beta toxin. Then you eat the sweet potatoes, which contain a chemical that turns off your natural trypsin protection. The beta toxin survives, accumulates, and just absolutely destroys the jejunum. It causes a necrotizing enteritis that is often fatal, especially in children. It's a tragic and amazing example of how diet and bacterial virulence can interact.
That is wild. All right, let's leave the tissue destroyers and move on to the nervous system. We have two Clostridia that target the nervous system, but they do it in completely opposite ways, creating this perfect dichotomy. First up, Clostridium tetani.
The agent of tetanus. Under the microscope, this one is beautiful in a terrifying way. It has a round terminal spore at the very end of the rod, which gives it the classic appearance of a drumstick or a tennis racket.
And this one is the brake line cutter.
That is the perfect metaphor for what it does. C. tetani produces a toxin called tetanospasmin.
And how does the infection start? This is the classic rusty nail scenario, right?
Right. You step on a nail, get a splinter, whatever. Spores from the soil enter the deep puncture wound. The wound closes up over the top, creating that perfect anaerobic pocket. The spores germinate, and they start to release tetanospasmin. Now, here's the journey the toxin takes.
Okay.
It's an AB neurotoxin. It binds to the motor neuron terminals right there at the site of the wound, but it doesn't act there. It hitches a ride inside the neuron.
Where is it going?
It travels via retrograde axonal transport. That means it moves backwards, up the axon, away from the muscle, all the way to the spinal cord.
And this travel time, that's what explains the incubation period.
Exactly. If the wound is on your foot, it has a long way to travel, so the incubation period might be a couple of weeks. If the wound is on your face, the path to the central nervous system is very short and symptoms can appear much faster.
Okay, so the toxin arrives at the spinal cord. What is its specific target there?
It exits the motor neuron and it enters the inhibitory interneurons, specifically cells called Renshaw cells. These cells are the brakes of your motor system. Under normal conditions, if you contract your bicep, the Renshaw cells release inhibitory neurotransmitters like glycine and GABA to tell the opposing muscle, the tricep, to relax. This allows for smooth, coordinated movement and prevents your muscles from fighting each other.
So the system relies on this perfect balance of go signals and stop signals.
Correct. Tetanospasmin is a zinc endopeptidase. It gets inside that Renshaw cell, and it cleaves a very specific protein called VMP, also known as synaptobrevin.
VMP is part of the snare complex. That's the docking mechanism for vesicles, isn't it?
Exactly. Think of the neurotransmitter vesicle as a cargo ship and the cell membrane as the dock. The snare proteins are the ropes that tie the ship to the dock, so the cargo, in this case glycine and GABA, can be unloaded. Tetanospasmin cuts the ropes.
The vesicle can't fuse with the membrane.
It can't. The inhibitory neurotransmitters are trapped inside the cell. The stop signal is never sent. The brakes are completely cut.
And if you cut the brakes on a car that has the gas pedal stuck to the floor, you get uncontrolled acceleration.
In the body, this means uncontrolled, simultaneous muscle contraction, spastic paralysis. Both the bicep and the tricep contract at the same time, fighting against each other.
Which leads to the classic horrifying symptoms.
It usually starts with trismus, or lockjaw. The masseter muscles in the jaw are very strong and have short neural pathways to the brainstem, so they often spasm first. The mouth clamps shut and can't be opened. Then it spreads. You get rhisis sardonicus, the sardonic smile.
An evil grin.
It's an eerie fixed grin because all the facial muscles are contracting at once. It looks like the Joker from Batman. And then you get opus sotenos.
The arched back.
Yes, the back muscles are much stronger than the abdominal muscles. When they both spasm uncontrollably, the back arches violently. The patient is often supported only by their head and their heels. It is incredibly painful, and the muscles can contract so violently they can actually fracture the patient's vertebrae.
That's just terrifying. And there's a neonatal version of this as well.
Yes, tetanus neonatorum. This is a major killer in many developing countries. It occurs when the umbilical cord is cut with a non-sterile instrument, or if, mud or animal dung is applied to the stump as a cultural poultice.
The infection enters through the umbilicus.
Right. And because a baby has such a short distance for the toxin to travel to the spinal cord, it hits fast and hard. The mortality rate is over 90%. It's a tragedy that is almost entirely preventable with clean birth practices and maternal vaccination.
So how do we treat tetanus? If the toxin is already inside the spinal cord, can we get it out?
No, that is the tragedy of it. The binding of the toxin to the neurons is irreversible. Once that toxin is inside, it's there until the neuron eventually degrades it and grows a new nerve terminal. That process can take weeks to months.
So the treatment is essentially supportive and preventative, stopping more damage.
It's based on 3 pillars. Pillar 1 is source control. You debride the wound to remove the bacteria so they stop making more toxin. Pillar 2 is antibiotics. Metronidazole is preferred to kill the bacteria.
Wait, why not penicillin? Penicillin is the classic drug for gram-positives.
This is a really nuanced but important point. Penicillin itself can act as a GABA antagonist. So if you give high doses of penicillin, you are theoretically blocking the very receptor that is already being starved of GABA. You could actually lower the seizure threshold and make the spasms worse. Metronidazole is safer in this context.
Okay, and the third pillar?
Neutralization. We give TIG tetanus immune globulin. These are preformed antibodies that bind the toxin. But, and this is absolutely crucial, they only neutralize the unbound toxin that's still floating around in the blood. They cannot reach the toxin that is already inside the nerve.
So TIG stops the progression of the disease, but it doesn't reverse the symptoms you already have.
Correct. And of course, the real solution is the vaccine.
The tetanus toxoid vaccine, part of the DTaP shot.
Exactly. It induces antibodies against the toxin, not against the bacteria. And one fascinating high-yield fact, getting clinical tetanus does not confer immunity.
Really? You can survive that whole ordeal and get it again?
Yes. The lethal dose of the toxin is so incredibly small that it kills you long before your immune system can even detect it to make a proper antibody response. It is lethal before it is immunogenic. So if a patient survives tetanus, you must vaccinate them afterwards to protect them in the future.
That's a wild concept. Okay, so tetanus cuts the brakes, leading to rigid spastic paralysis. Now let's look at the flip side of the coin. Clostridium botulinum, the gas line blocker.
See botulinum. This causes botulism, and it leads to a flaccid paralysis. You go limp.
How does it manage that?
Well, it also has a toxin, botulinum toxin, and in fact, this is the most potent toxin known to science. The lethal dose for a human is measured in nanograms. It's unbelievably powerful.
So where does this one strike?
Tetanus toxin traveled all the way upstream to the spinal cord. Botulinum toxin is, let's say, lazy. It stays at the peripheral neuromuscular junction, right where the motor nerve meets the muscle fiber.
And what does he do there?
It is also a zinc endopeptidase that cuts snare proteins. But instead of hitting the inhibitory neurons in the spine, it hits the excitatory motor neurons out in the periphery, and it prevents the release of acetylcholine.
Acetylcholine is the go signal for muscles. It's the neurotransmitter that makes them contract.
Exactly. So the brain says move, the electrical impulse travels all the way down the nerve, but when it gets to the terminal, the acetylcholine vesicles can't dock because the snare proteins have been cut. The signal just dies right there.
No gas gets to the engine.
Perfect analogy. The muscle never receives the instruction to contract,
Resulting in a flaccid paralysis.
Yes. And there's a slight difference in which snare protein is targeted based on the toxin type. Types A and E cleave a protein called SNAM25. Type B cleaves VMAMP, the same one as tetanus, but in a totally different location. But the end result is the same. No acetylcholine release.
There are three main clinical ways you can get this. The first is classic foodborne botulism, and this is an intoxication, not an infection.
Right. Yes, exactly, meaning you eat the preformed toxin. This is the classic home canning scenario. You have some green beans or corn, which are alkaline vegetables. Botulism spores hate acid, so tomato sauce is usually safe, but things like green beans are risky. You can them at home, but maybe your water bath isn't hot enough or pressurized enough to kill the spores.
The jar sits on the shelf for months. The spores germinate in the anaerobic vacuum of the jar, and they produce the toxin.
Months later, you open the jar. The food looks fine, it smells fine, it tastes fine, you eat it. You are ingesting pure, preformed poison. It gets absorbed from the gut into the blood, and it circulates, hitting all the peripheral nerves.
And the symptoms, how does this present?
It presents as a bilateral descending paralysis,
Yeah.
top down. It starts with the cranial nerves because they have the shortest path. The 4 Ds are the classic mnemonic: diplopia, double vision; dysarthria, trouble speaking; dysphagia, trouble swallowing; and dyspnea, trouble breathing.
What does that look like at the bedside?
The patient looks at you, and their eyelids are drooping. That's ptosis. They try to follow your finger with their eyes, but their eyes don't move together, so they see double. Their speech sounds slurred and thick, like they're drunk. But, and this is the real horror of it, they have a clear sensorium. They are fully awake and aware of everything that's happening. They just can't move.
And as the paralysis descends, it hits the diaphragm.
And they stop breathing. That is how it kills you. Respiratory failure.
Okay, the second form is infant botulism. And this is actually the most common form in the U.S. How is this different?
This is an infection, not just an intoxication. In this case, a baby ingests the spores, not the preformed toxin.
And where would a baby get the spores from?
Dust, soil, or famously honey. Honey is a raw, natural product, and it can often contain botulism spores.
And this is why we have the strict rule. No honey for any child before the age of 1.
Correct. If an adult or an older child eats honey, our mature microbiome, our gut police force, outcompetes the spores. They just pass through harmlessly. But a baby's microbiome is immature. There are lots of empty apartments. The spores can germinate in the baby's gut, they colonize it, and they produce the toxin in vivo.
And what are the symptoms in a baby?
It's much more subtle than in adults. Constipation is usually the very first sign, and it's often missed or overlooked. Then you start to see the floppy baby syndrome. They have a weak cry, poor feeding because they can't suckle effectively due to muscle weakness, and they lose their head control. They feel like a rag doll when you pick them up.
And the third form is wound botulism.
This one is strongly associated with injection drug use, specifically with black tar heroin. The heroin itself is contaminated with spores from the soil. Users will inject it into the muscle or under the skin, a practice called skin popping. This creates an anaerobic abscess.
A perfect incubator.
Exactly. The bacteria grow in the abscess and produce toxin, which then disseminates into the bloodstream. It presents just like foodborne botulism, a descending paralysis, but it often comes with a fever and a high white blood cell count because of the underlying wound infection.
How do we diagnose botulism? It seems like you'd have to be really quick.
It's very tricky. Routine labs are all normal. A spinal tap is normal, which helps distinguish it from Guillain-Barré syndrome. You have to find the toxin itself. The gold standard, historically, is the mouse bioassay.
That sounds incredibly old school.
It is. You literally inject a sample of the patient's serum or stool into a mouse. If the mouse develops the classic signs of botulism and dies, and then a second mouse that was pre-treated with antitoxin survives, you have your diagnosis. Nowadays, we are moving toward faster PCR and mass spectrometry methods, but the bioassay is still considered the definitive confirmation in many textbooks.
And the treatment?
Number 1, 2, and 3 is ventilatory support. You have to breathe for them, potentially for weeks or months, and you give antitoxin. For adults, there's a heptavalent antitoxin derived from horses. For babies, we use a human-derived product called Big 4, or botulism immune globulin, to avoid allergic reactions.
But again, like with tetanus, the antitoxin only binds the circulating toxin that hasn't bound to a nerve yet.
Right. It stops the paralysis from getting any worse. But for the nerves that are already blocked, you just have to wait. The nerve has to physically regenerate new terminals to restore the connection to the muscle, and that can take a very, very long time.
Before we wrap up the main villains, let's briefly touch on the other Clostridia. The opportunists, there are a couple that are very high yield for boards.
We absolutely must mention Clostridium septicum.
C. septicum, the red flag organism.
This is the cause of non-traumatic gas gangrene. If a patient comes into the hospital with myonecrosis muscle death, but they have no history of trauma, no wound, no reason to have it, and you find C. septicum growing in their blood culture, you need to panic. But not just about the infection.
What else are we worried about?
Colon cancer or leukemia. There is a massively strong association. The thinking is that an occult tumor in the colon creates these microulcerations or perforations in the gut wall. This allows C. septicum, which is unusually aerotolerant for a Clostridium, to translocate from the gut into the bloodstream and then seed a distant muscle.
So C. septicum bacteremia is basically a paraneoplastic sign of an underlying cancer.
Exactly. If the patient survives the septic shock from the infection, they need a colonoscopy immediately.
And the last one on our list, Clostridium sordelli.
This one is just tragic. It causes a fatal toxic shock syndrome. It's most famously associated with childbirth or medically induced abortions, particularly with the use of mifepristone and misoprostol.
What makes it so distinct clinically?
It presents with a massive capillary leak syndrome. The patient has profound, severe edema, massive swelling everywhere. They get hemoconcentration where their blood becomes thick like sludge because all the plasma has leaked out. And they have an extremely high white cell count, 50,000, 100,000. It's called a leukemoid reaction. But the real kicker is they are often afebrile.
No fever in a toxic shock syndrome.
No fever. The toxin seems to suppress the inflammatory cytokine response even while it's destroying the body. And this is so dangerous because the patient doesn't look septic in the classic sense. They aren't hot and flush. They can be in profound shock before anyone realizes how sick they are, and by then it's often too late.
Wow. Okay, let's try to synthesize all of this.
Yeah.
We have this family of bacteria that hide in bunkers, the spores, and then use these incredibly potent chemical weapons, the toxins, to do all their dirty work.
Right, and you can see the pattern. C. diff is the squatter that waits for antibiotics to clear the room, then it destroys the gut scaffolding with toxin B.
C. perfringens is the speed demon that melts flesh with its alpha toxin, the lucithinase.
C. tetani travels retrograde to the spine and cuts the brakes. It cleaves snares in the Renshaw cells, and that causes rigidity.
And C. botulinum stays peripheral and cuts the gas-line cleaving snares at the neuromuscular junction, which causes floppiness.
And in all of these cases, recognizing the clinical pattern quickly is far more important than waiting for a lab culture.
Absolutely. These are clinical diagnoses first and foremost. If you wait for lab confirmation of botulism, your patient will have stopped breathing days ago. You have to treat based on suspicion.
You do.
All right, I think our listeners are ready to test their knowledge. Let's do a rapid-fire quiz to really lock this in. I'll ask the question, we'll pause for just a second so you at home can think, and then our expert will give the answer and the why.
Let's do it.
Okay, question 1. A patient arrives in the ER with locked jaw and severe back spasms. The causative agent produces a toxin that cleaves snare proteins. Which specific neurotransmitter release is being blocked?
The answer is glycine, or GABA. And the why is, this is tetanus. Tetanospasmin travels to the inhibitory interneurons, the Renshaw cells, in the spinal cord. It blocks the release of the brake signals GABA and glycine, which leads to unopposed excitation and rigid paralysis.
Question 2. A 6-month-old infant presents with constipation and flaccid paralysis, the classic floppy baby. The parents admit to sweetening the baby's formula with honey. What is the mechanism of this disease acquisition?
The answer is ingestion of spores, which then germinate and produce toxin in vivo. The why is, this is infant botulism. It is a critical distinction. Unlike adults who typically get sick from preformed toxin in canned food, infants get sick because they eat the spores. Their immature gut microbiome can't stop those spores from germinating and producing the toxin directly inside the gut.
Question 3. A patient who is recently on clindamycin develops profuse watery diarrhea. A colonoscopy reveals characteristic white plaques on the colon wall. Which specific virulence factor is primarily responsible for the disruption of the cytoskeleton in the epithelial cells?
The answer is toxin B, the cytotoxin, the why. This is C. diff. While toxin A is the enterotoxin that attracts neutrophils and causes the fluid loss, it is toxin B that is the true cytotoxin. It glycosylates the Rho GTpases, which leads to the depolymerization of actin and the cell death that forms the pseudomembrane.
Question 4. You are looking at a Gram stain from a rapidly progressing necrotic wound. You see large rectangular Gram-positive rods, but you notice a distinct and eerie absence of neutrophils. What toxin is responsible for this finding?
That would be the alpha toxin, lecithinase. The why is, this is C. perfringens gas gangrene. The alpha toxin is a phospholipase that lyses cell membranes. It destroys the white blood cells as soon as they arrive at the site of infection, which explains why you don't see pus or neutrophils on the slide, despite the incredibly severe infection.
Question 5. A 45-year-old male presents with spontaneous gas gangrene in his thigh. He has had no recent trauma. His blood cultures grow Clostridium septicum. What critical underlying condition might you immediately rule out?
The answer is colon cancer, or another GI malignancy like leukemia. The why is that C. septicum is an air-tolerant opportunist that can translocate across the bowel wall, often through microperforations caused by a tumor. Its presence in the blood is a sentinel sign for an underlying malignancy.
All right, last one. Question 6. Regarding C. perfringens food poisoning, the symptoms are caused by a toxin that is produced in which location?
The answer is in the patient's small intestine. The why here is crucial. You ingest the vegetative bacteria from the contaminated food. The enterotoxin itself is only produced inside you, as the bacteria undergo the process of sporulation in the alkaline environment of your small intestine.
Well, that was quite a journey through the anaerobic underworld. Before we sign off, do you have a final thought for us to chew on?
I do. It's about the duality of nature, I suppose. We spent a lot of time talking about how unbelievably deadly botulinum toxin is, the most lethal substance known to man, and yet millions of people voluntarily inject it into their faces every single year. Exactly. Botox is just a purified and incredibly diluted form of botulinum toxin A. The exact same molecule that causes flaccid paralysis and death is used to paralyze facial muscles to smooth out wrinkles or to treat migraines and severe muscle spasms in conditions like cerebral palsy.
It's the ultimate testament to the idea that the dose makes the poison.
It really is. We have taken the beast, the most powerful biological weapon on the planet, and we have tamed it. Mostly. It's a wild thought.
It is a wild thought.
Yeah.
A biological weapon that became a cosmetic blockbuster. Thank you all for listening. If you want to consolidate this information, go read the clinical case boxes in Murray Chapter 30, specifically the ones for tetanus and botulism. They do a great job of mapping out the timeline of symptoms.
And remember, if it smells like a barnyard, wash your hands with soap and water. Alcohol gel does not kill spores.
A great tip to end on. See you next time on The Deep Dive.